Hello everyone. I'm myself Yashwant, research scholar at Department of Bioscience and Bioengineering at IIT Guwahati. So today I'm going to explain about kinetics of cell growth and substrate utilization. Microbial growth is a result of change in cell number and the cell size. So once we inoculate a culture such as bacteria or yeast or any protozoa or something inside the culture medium. So cells uptake the nutrients, there will be doubling and division of cells, so hence cell number increases and there is a cell size also increases and because of the mass accumulation. During this time there is a conversion of the nutrients such as carbon sources such as glucose, nitrogen such as various ammonia, so many other nitrogen sources, yeast extract and phosphorus. This will be converted biological compounds which are useful for the energy production basically. Ultimately ATP is produced and synthesis of the end product or the desired product. So the growth rate depends on the number of cells inoculated, stage of the inoculum, nutrients present in the medium and culture conditions. The nutrients present already I have discussed, it is macronutrients and micronutrients, and the culture conditions is basically temperature, pH and agitation required for the culture growth. So typical when we consider typical growth curve. So if we consider bacteria or yeast, it follows the typical growth. So if it's a batch culture basically. So the growth of the culture is represented as a growth curve. So it is divided into four steps. One is the lag phase, exponential phase, stationary phase, and death phase. In lag phase, the cells adapt to the new environment, showing little to no division. In exponential phase, cells grow and divide rapidly at a constant rate. In stationary phase, the growth plateaus due to nutrient depletion or waste accumulation. That means there is no net growth rate. In death phase, cells die at an accelerated rate due to adverse conditions. In lag phase, there is no increase in cell number as I told earlier, but there is an increase in the mass because there is no division, there is uptake of nutrients at a lower rate. It's a period of adaptation of new cells to the new environment. Basically, is the temperature, media composition, pH, everything should adjust so that at the optimal condition the cells will grow properly. So the culture conditions and the media composition will decide the length of the life phase. Now, sometimes there is multiple carbon sources that result in multiple lag phases. For example, sometimes the cells utilize lower concentration of glucose and glycerol is utilized. So first cell utilize the glucose and then it will adjust the metabolic activities to utilize the glycerol present in the second carbon source present in the medium. This kind of growth is called as diauxic growth. So since sometimes we use complex sources such as in the case of lignocellulose biomass treatment, we extract the after the pre-treatment we extract the medium and that contains that is rich in xylose, glucose, etc. So similarly, crude glycerol contains amount of glucose, sucrose, so constable liquor. So these compositions are very complex. So this time there is a diauxic growth will occur. Log phase is the exponential phase of the growth phase. The cell growth rate is higher in log phase with increase in cell number and cell mass is a period of balanced growth. Balanced growth means the cells uptake the nutrients, they increases linearly or exponentially linear. Hence, there is a balanced growth. The composition of biomass remains constant throughout the growth period. So unbalanced growth may be because of the limitation of some nutrients or something. This is a rate limiting nutrient or the accumulation of toxic products result in the deceleration phase or the decline phase after the log phase. So kinetics of log phase. The kinetics of growth phase or the exponential phase for first order reaction. Here the rate of the biomass synthesis is proportional to the rate of biomass present. So if we consider the equation 1, Rx is equal to mu into x. Rx is the volumetric rate of biomass production which has a unit of kg per meter cube per hour, comma, x is mass of cells kg per meter cube and mu is specific growth rate per hour. Rx is equal to dx by dt. Combining equation 1 and 2, Rx is equal to dx by dt is equal to mu x, integrating over a period of time from 0 time initial time with a biomass concentration x naught and to the time t at with a biomass concentration x. So we will get the equation ln x by x naught is equal to mu into t. X by x naught is equal to exponential of mu t. After plotting ln x by x naught versus time, we will get a slope. We will get a straight line with a slope mu, which is follow the first order reaction. Hence, we can calculate the specific growth rate by plotting ln x by x naught versus time. And this is also this equation can also be useful in calculating the doubling time of a microorganism. In stationary phase, after the log phase, there is a decline phase or cells dissolution phase. Cells started adjusting. Then, because of this, there is a rate of net growth rate is equal to net death rate. Hence, there is net growth rate is zero. Because of this, ds by dt or mu is equal to zero. After the stationary phase, the cells started entering the decline phase or the death phase. Here, the death phase is greater than the growth rate. Hence, due to the consumption of most of the nutrients present in the medium, the accumulation of toxic excretory products will occur, which won't support the growth. Instead of that, it supports death and the culture crashes. Kinetics of cell death. Similar to the kinetics of the cell growth or the growth phase, here log phase, the death phase also has a kinetics which follows the first order reaction. Here, the rate of cell death is greater than the rate of cell growth during the log phase, where if we consider the equation RD is equal to KD into N, where RD is the rate of cell death, N is the number of viable cells, and KD is a specific death constant. RD is equal to minus dN by dt is equal to KD into N, where KD is the specific death constant and RD is the rate of cell death and N is the number of viable cells at time t. After integrating, when n is equal to n naught into e to the power minus kd into t, after taking the natural logarithm, ln n is equal to ln n naught minus kd into t. If that follows the first order kinetics, a plot of ln n minus ln n naught versus time always gives the straight line with a slope minus kd, which is negatively correlate. So, cell death rate also follows the kinetics of first order rate kinetics. Similarly, the log phase or the growth phase also follows the first order kinetics. So let us look into the summary of the growth kinetics. The lag phase, there is adaptation of medium, adaptation of cells to the medium, and there is no growth. Specific growth rate is zero. In growth phase, there is a maximum growth, hence mu is equal to mu max. In decline phase, the growth slows down and hence mu is less than mu max. Stationary phase, growth ceases, mu equals to 0. Death phase, loss of self viability, mu less than 0. So we have learned about the growth curve, a typical growth curve which has four phases. After that, so during this time, especially during the log phase, the substrate utilization is very high. So how do you utilize substrate? So the cells will utilize the substrate present in the culture. So the specific growth rate is dependent on substrate concentrations present in the medium, basically. That sometimes the slow growth will occur because of there may be carbon limitation or nitrogen limitation may occur, hence it may slow the growth, and this is called growth limiting step or the growth limiting substrate. So this growth rate can be represented similar to the enzymatic kinetics like Michaelis-Menten equation. We have something called monod equation which gives the information of specific growth rate. So here we can get specific growth rate mu is equal to mu max into S divided by Ks into S. S is a concentration of growth limiting substrate and mu max is maximum specific growth rate and Ks is substrate concentration where maximum mu reaches half the maximum specific growth. That means when the substrate concentration is very excess, when S is excess, S is very much greater than Ks, hence mu becomes mu max, that is mu is independent of substrate concentration. That is, there is no substrate inhibition. So here, when Ks is also a substrate concentration, if substitute Ks is equal to S, in the monod equation, mu becomes mu max by 2. That means Ks is substrate concentration at which the rate of the reaction is half of the maximum specific growth rate. So we can observe the same in the plot. This monod equation, using this monod equation, once again calculate the growth rate, specific growth rate, that is maximum specific growth rate, and Ks and mu max. So after reciprocating the monod equation, we get the equation as 1 by mu is equal to Ks plus S divided by mu max by S. 1. This simplifying step further, the equation simplifies to 1 by mu is equal to Ks by mu max into 1 by S plus 1 by mu max. This is equal in the form of first order equation y is equal to mx plus c, where m is nothing but the slope, and slope is equal to Ks by mu max, and c is the y-intercept is 1 by mu max. After once we plot 1 by mu versus 1 by s, we will get a straight line which for first order kinetics. Here, the after extracting the slope, we can calculate the mu max by y-intercept, and Ks we can calculate using slope as well as x-intercept. So this equation is very much important in the determining the rate of maximum growth rate of the yeast or bacteria, and similarly this equation also applies in the enzyme kinetics. So once a substrate is utilized, the cell is going to produce the biomass along with the product. So how to what extent the substrate supplied is converted efficiently to a biomass or product is determined or expressed in growth yield coefficient. It is defined as unit amount of cell mass produced per unit amount of substrate consumed, that is nothing but dx by dt is equal to 1 by y into dx by dt. Y is cell yield, nothing but the yield coefficient, that is a gram of biomass produced per gram of substrate consumed, where delta x is mass or the moles of cell biomass produced into delta s is mass or moles of substrate consumed, where y x by x is equal to delta s is nothing but x2 minus x1 divided by s2 minus s1. The yield is theoretical, the yield can reach the maximum value of 1. If that means the substrate supplied is completely converted to the biomass or the product. In the case of ethanol conversion by Saccharomyces, if you supply 2 gram per liter of glucose, the ethanol produces 1 gram per liter. That is 50% theoretical yield. This is called theoretical yield of ethanol, and there is something called experimental yield, that is always the real yield, which is based on the experiment or the cultural conditions. So similar like this, we can analyze for various bioreactions, we can analyze the growth yield coefficient. So thank you so much. Please, if you have any queries, you can ask.